Evolutionists have consistently claimed life can arise by chance from non-living elements based on supposed proofs, such as the famous Miller-Urey experiment. In 1953, Stanley Miller and his mentor, Harold Urey, simulated a highly speculative early Earth primordial soup and then declared they had created life from this mixture. However, nothing could be further from reality. In fact, not only did they fail to create a complex living creature, but most of the components produced by their experiments was destructive tar, which would have effectively eliminated any possibility of early life. As former evolutionist Gary Parker discovered, what Miller actually produced was a seething brew of potent poisons that would absolutely destroy any hope for the chemical evolution of life. The true findings of this well-known experiment are often ignored by evolutionists who have been successful in brainwashing several generations of unsuspecting biology students into believing science has created life from dead particles, making evolution from a primordial soup appear feasible. In reality, the Miller-Urey experiments were highly unscientific and unsuccessful attempts to create life from non-life. All truly scientific observation has concluded life does not arise by chance from non-life. Molecular biophysicist Cornelius Hunter confirmed it is a fundamental tenet of molecular biology that life comes from life, and evolutionary origin of life research is nowhere close to proving this tenet wrong. As exemplified by recent cloning efforts, the formation of life from non-living components such as amino acids requires organization by an intelligent being. Evolutionary arguments for the chance origination of life from non-intelligent and unpredictable natural forces disintegrate at this point, but for the sake of thoroughness, we will temporarily imagine, together with evolutionists, that all life-specific amino acids somehow arose by pure chance within the disorganized primordial soup of primitive Earth, which came from a chaotic primeval explosion bursting from nothing and nowhere. We're now going to see why, for evolutionists, these statistical and biological problems are powerful enough to kill their dying theory. Amino acids are protein building blocks, which are primary biological components. Average proteins are exceedingly complex chemical structures containing 400 precisely sequenced amino acids. Since 20 different varieties exist, 100 amino acids can be arranged in 10 to the 130th power different ways. To appreciate how large this number is, consider there are only 10 to the 84th power of subatomic particles in the entire universe. It has been calculated by statisticians that if just 100 amino acids were arranged randomly 1 trillion times per second for 4.6 billion years, then the probability of them being arranged once in an order capable of producing just one protein is 0. Adding to this impossibility, we must remember the average protein contains 400 instead of only 100 amino acids, and even the smallest living creatures feature hundreds of proteins, which must also be exactly ordered to manufacture DNA. Evolutionists claim the first living creature springing from primordial soup was a single-celled bacterium possessing approximately 100,000 base pairs of DNA nucleotides and 10,000 amino acids with at least 100 functional protein chains. Since it would be impossible for even one of the proteins within this simple bacterium to form by pure chance over 4.6 billion years, we must conclude evolutionary theory is unfounded. The issue of chirality presents further statistical difficulties for evolutionists' hypothetical first bacterium. Chirality explains the fact that all nucleotides in a DNA or RNA chain must have a specific molecular conformation for the chain to work. All nucleotides must be located on the right side of a DNA chain, and all 20 amino acid types must be arranged in a specific order on the left side for a protein to function. Within an initial theoretical cell, all 10,000 or so diverse amino acids in the approximately 100 functional protein chains must have suddenly combined on the left side at exactly the right time in a precise sequence, and all 100,000 nucleotides must have combined in a specific right-handed sequence at the perfect time to form a DNA molecule able to direct cellular activity. The chance collection of 100,000 properly oriented nucleotides is similar to the probability of a coin being flipped onto its head 100,000 consecutive times, while creating the specific sequence of amino acids needed for life would require flipping 10,000 tails in a row. Simultaneous achievement of a precise nucleotide and amino acid conformation is equal to the probability of someone correctly calling the side of 110,000 consecutive coin flips. The odds of this happening are a staggering 1 in 10 to the 33,113th power. This is the same chance one would have of winning 4,700 state lotteries in a row with only a single ticket for each contest. The probability of accurately sequencing all 110,000 nucleotides and amino acids in a primordial soup is also equivalent to the chance that a blindfolded person would successfully choose a single marked particle from nearly 400 galaxies as large as the Milky Way. Formation of the required chirality in a primordial soup or any other environment, even if all the necessary parts are present, is statistically impossible. While chirality alone is an insurmountable problem for evolutionists, there are still other factors to consider in the origination of a supposedly simple cell. Specifically, since only 20 of the 80 amino acids on Earth are life-specific, the correct amino acids must have met in an alleged primordial soup. The probability of this private conglomeration occurring by pure chance in the first cell is 1 in 10 to the 6,021st power. Also, since 20 different life-specific amino acids exist, there would be a 1 in 20 chance of the right amino acid collecting in the right spot on each of more than 100 protein chains individually containing several hundred amino acids. Chirality only considered the orientation of amino acids, not the specific sequence. The probability of each life-specific amino acid randomly appearing in the proper spot on the correct chain is 1 in 10 to the 13,010th power. Additionally, the right material for each gene must have gathered so that all genes were capable of performing its necessary body function. The simplest single-cell bacterium contains between 265 to 350 genes vital to life. With 100,000 base pairs, the most basic bacterium requires about 377 base pairs to be correctly sequenced per gene. The chance of this happening in 265 genes is 1 in 10 to the 60,155th power. Further, for DNA to properly direct the life function of a cell, all genes must be precisely sequenced on the DNA chain. The chance of this occurring with 265 genes is 1 in 10 to the 528th power. Before adding all these probabilities, it's important to realize scientists regard anything with less than 1 chance in 10 to the 50th power of occurring randomly as impossible or absurd. With that in mind, we should be astounded by the fact there is a mere 1 in 10 to the 112,827th power chance the simplest conceivable creature could randomly arise from a hypothetical primordial soup. This is the same probability someone would have of winning 16,119 state lotteries in a row with a single ticket for each, or choosing, while blindfolded, a single marked electron for more than 1,300 Milky Way-sized universes.